Right. Okay, what you can do, you can even use a single peptide. Just take a single peptide and immunize that. What kind of problem will it have? Same problem, right? It's exactly the same problem, right? And actually even lower immune response. Virus cells. What else one can do? You can do like virus-like particle as a cargo, and then you can just modify it. You can take just a virus. This is a virus, right? And then you can pseudotype it. So that's an important term. You will see it many times, like again and again. Look at this. So P is silent, pseudotyping. You see that? You can write it down. Okay. Although my writing is, you need to get used to that. It's a little bit. Okay, pseudotyping. Okay, what is pseudotyping? Is when you repurpose the antigens of the virus in order to have different properties. Whatever you like, you can repurpose that to bind to a different receptor on the cell, so that they will bind to a different cell. They will enter different cells, or you can pseudotype it in a way that it can be conjugated with some protein or mucus. Whatever you wish. So this is called pseudotyping. Everything which is unnatural is called pseudotyping. Okay. Right. You can enter it within the membrane. That's also possible. Not conjugated, but just insert it within the membrane. You can use permissive insertion site. So what is a permissive insertion site? You have some structure of the protein, and then you need to use some binding tools, and later also validate that in vitro, that some specific portion of the protein is not critical for its activity. Or it could be related to the protein. It could be structure. Yeah, so the protein is just a structural protein, so it is robust. It holds the whole structure together, right? Or it could be an enzyme, right? Enzymatic, or it could be a receptor. It could be some sites where it binds. So the permissive type means that you can bioconjugate, so you can make a fusion at that point, so that it doesn't disturb the function of the protein. And then, yes, you can just gain some whatever proteins you wish. Okay? So that is called the permissive insertion site. Okay, and many other points. So you can fill the viral particle with something. You can use chemistry and so on and so on. So like both chemical modification or genetic engineering and so on and so on. Here you can see those examples. Okay. And in the end, RNA vaccines, which are usually delivered in liposome-like nanoparticles, and those, as well as these either live vaccine. There are also live vaccines. So sometimes you can use also live vaccine. The virus, which is intact, and it gets inside the cell, and it delivers its cargo, as you can see here. Yeah, so the virus is of another type carrying the antigen, and those will go inside the cell. They will internalize, and they will induce also the T products. So those RNA vaccine or viruses which get inside the cell are the most important in terms of the effectivity of the immune response. Okay, okay, so we're talking about the viruses and also like all these diseases. Yeah, so there are indeed many diseases which can be caused by viruses, and that's why the Peter Medawar coined them viruses is a piece of nucleic acid surrounded by bad news. That's a little bit ironic because actually there are much more viruses which cooperate and co-evolve with us, and they are either indifferent for us or they are actually even helping us to develop some of our properties. If you look at, we'll talk about that a little bit later, that non-coding RNAs, which were really boosted from like in the time which we, when these hominids, the primates, started to develop those eggs, right, and humans. So we have much more of those related transplants, so those ancient viruses which jump from place to place in order to achieve to transfer some of the genetic material. This means that also to duplicate the genes and to make some of those copies of the genes to be taken out of the evolutionary regression, but we have one copy, but the second copy not anymore under the evolutionary regression, so it can evolve and so that we can gain new functions. And overall, so apart from that, apart from these lentiviruses, so we have also gained a lot of different functions in the viruses. So many of them really live together with us without causing any harm, or some of them they also become kind of they are at first kind of more when we just gain them from nature, they were kind of really harmful, but later they become less harmful. So we also co-evolve together with that. So overall, I would like to stress that it is a little bit ironic, right here. So what this Peter Medawar was saying that it is mostly what we are talking about when we are talking about viruses. It's about something disease and so on and so on. But it is actually not true. You will see what I'm talking about. But now we'll have a short break, so we'll continue in 10 minutes. By the way, is it cold or warm? Is it comfortable for you? Or I can switch on the AC if needed, so to make it a little bit warmer if needed. Huh? Should we? Okay, just let me know, okay, if needed. It's okay. Okay. Okay, so let's continue. We were talking about how ubiquitous a virus is and how we actually live together with them, even if we don't think about that. So the efficiency of replication by viruses is so high that a single virus can infect the can produce so many viruses which could be enough for the to infect the whole human population. For example, if we are talking about the human influenza virus, so it can so a single person can produce as many viruses as it is needed to infect the whole population of the Earth, for example. Yeah, and current estimates of the number of individual viruses suggest that they exceed the number of the stars in the universe. So it's about 10 in the power of 23. So it's really, really huge. Let me show you another example. Yeah. So near the town of Chihuahua in 2009, they found a giant crystal cave. It remained isolated from the rest of the world, completely isolated for 26 million years. And since it was close to the magnetic sources and close to the volcano, so the conditions were so that all those giant crystals formed. Yeah, so it was absolutely out of the range of any kind of living creatures for so many years, right? So yet every drop of water inside contained 200 million viruses. Can you imagine that? So, and we're talking only about the completely isolated system, right? So at the same year, 2009, so Daniel Wilner, who was a scientist, she decided to make a project, and in order to check the people with cystic fibrosis, so with a lung disease, and healthy individuals, in order to check. So at that time, it was considered that, hey, so the healthy individuals, the saliva and all the internal organs, they are survive. They are devoid of any kind of foreign agents and so on. So, and she was really surprised that actually it doesn't matter whether it was like the lung disease or healthy individual. In any of them, she found 174 different species of viruses, many of them which were not even discovered. So you can really imagine. So like if you think about how many of the bacteriophages we face everywhere in the soil, in the water, in the air. So this is of course like really mind-blowing. So the majority
viruses what we have in our environment are bacteriophages, or they're called also phages or bacteriophages. What is a phage or bacteriophage? Is a virus which infects the bacteria. Okay, so that's the only difference. So generally, that's how they call it. So what is a phage or bacteriophage? Is a virus which infects prokaryotes, right? So it's really remarkable that actually viruses, they can also impact our environment and also global climate, because those bacteriophages which can infect cyanobacteria within the ocean, and they can control how much of the CO2 is being captured or being released. This per se could also like regulate the levels of the temperatures, and so this is so what I want to say is like viruses are really everywhere, and we confront with them all the time, and it is not only about diseases, it's also about like living hand by hand with them and also being co-evolved with them. And later you will see that viruses arguably are also something which we ourselves derive from, because I will tell you a little bit later that in the end, like whatever is our forefather is arguably like just a molecule, one molecule of RNA. But we'll talk about that a little bit later. Right, so we talked about the high efficiency of replication. We talked about the influenza virus, which can infect the whole human population, about the overall estimation of the viruses. And if one could take all the viruses, all the bacteriophages and viruses from the ocean and make it a single line, just one after another in a single line, then this length would exceed the distance from the 60 galaxies from so it is really enormous. Like if we are talking about how ubiquitous are the viruses. I hope I have convinced you. And now let us dive into a fascinating world of the prominent researchers who devoted their life to studying viruses. So let me first look at whether I still see the Zoom, because in one of my devices I don't have the Zoom anymore. Okay, so it's reconnecting. That's fine. Okay. So first, of course, Edward Jenner. He was probably the first person, the first doctor who was so curious and whom we all actually the fact that we managed to eradicate smallpox. Smallpox is a deadly disease, really deadly disease. It's a very infectious one, and it is caused by a virus called variola. So variola, which belongs to the pox viruses, the infection rate is really high. So it is very infectious and also very deadly. Okay, so a very high percentage of deaths, and it was known since thousands of years, and also since really ancient times, people knew that if one takes some sample from the fistula, from those those toxins, from this from those wounds of those dead people, and variolate, that is what is called variolate, inject under the skin some of this sample. Some people develop immunity. This was called variolation. This was very common. It was done both in China, also in ancient India. So people knew that for a long time. If they are lucky not to die from that, and that's about good, then some of the people that so Edward Jenner, when he was a kid, he was also very related to himself. So he was immune against smallpox. And he was a very curious doctor, and he thought that in some of the women who were dealing with cows, and they were taking milk, they were taking care of them. They were also immune against smallpox very often. It turns out that cows, they had also some analogous disease, which was this cow pox. And it was pretty mild. So it was some kind of small lunch, but they were not dying from that. And even some people were also having some kind of rush for that. So it's basically there was some mild form of disease. And we saw that those people who had contact with those kind of with this kind of cows which had this disease, which in Latin, the cows, that's like those viruses is called vaccinia virus. Okay, so that one from the smallpox is called variola, and that one is vaccinia virus. Okay, from the word cow. Okay, black cow. So they saw that you can do not variolation, but you can do vaccination. So what they did is they took this kind of sample of this pustula from this cow pox and then injected under the skin, and those people were also immune without any kind of adverse effects, so they were not dying. Because some variation that could be dangerous because you just inject the smallpox and let's see from that, right? And that's what he did. So he first injected to several people, and then indeed it turned out to be very effective. And then Great Britain wide, so they induced this kind of obligatory vaccination, and it took. As you can see, so he did that in 1798, and we actually eradicated the virus in 1979. So it took really so long to enforce the vaccination worldwide, but yet, so it is the first virus which we have completely eradicated from the human population. So there are still some samples of the smallpox which are available in the US and in Russia, and some like super like biotech compartments. So just in case, like, okay, we need to continue some research, just in case, like, we will be able to contain the virus or the virus later at some point. But otherwise, it's eradicated from human population. The second one, which probably will be eradicated soon, could be poliomyelitis. So we are very close to that. We're not yet there, but we're very close. So there are still some poliomyelitis outside, but that's it. So far, as you can see, we were not very successful. We have managed to eradicate only one of these deadly diseases, this smallpox. Okay. Okay. So then, who else? Okay, Louis Pasteur. Who, as I told you, so he developed this rabies vaccine. We talked about that. Remember, so about the rabies in the past until it's getting inactivated. Although he didn't know that it is a virus, and also he managed to develop what this porcelain filter to divide cellular from non-cellular. Okay, and then due to him, Adolf Meyer, he managed to filter through this agent. So actually, Adolf Meyer didn't filter. He saw that if one take the washout from this leaves of the dental mosaic virus infected plants and pass it to another one, well, they got infected. So it's just that might be actually this some infectious agent, right? Not yet knowing whether it is like cellular or subcellular. And then Dmitry Ivanovsky, he suggested, okay, so he filtered that through and saw that it's actually not cellular. So he suggested that it could be something non-cellular. And then it was later also confirmed by Martinus Beijering, who also coined the term virus. Okay, so that's what he thought. That is like non-cellular infecting agent. And then later, Paul Frosch and Frédéric Lefleur, who were they were students of Robert Koch. So they managed also to show that for the foot and mouth disease, like very similar. So they also filtered it through and also showed that it's also non-cellular agents. Then later, Carlos Finlay, in 1881, he saw that yellow fever transmission was attributed to mosquitoes. And then Walter Reed, who was a doctor, he managed to filter this agent and also show that it is also having the non-cellular nature. Then there were several other discoveries by showing that there was some leukemia virus which was discovered also in the beginning of the 90s, but the very first Nobel Prize was given to Northrop and Stanley for the crystallization of the tuberculosis virus, because it was really surprising how can the virus be crystallized, or something infectious can have a crystal structure, right? So this was for the very first time, and it was as late as 1946. Although already in 1911 there was Raus who managed to discover the sarcoma virus. So the virus which can cause the solid tumor. It was not the first tumor-inducing virus, as I told you before. So they were previously they had discovered that there were some viruses which were causing the leukemia, but for the solid tumor, it's sarcoma. That was the very first one, and people didn't believe that. People really were had a really big problem with that. So he discovered that in 1911, but he got his Nobel Prize in 1966. 55 years later, you know, so people really didn't believe that. Okay, so Max Tyler, we talked about that. So he managed to discover the yellow fever vaccine, develop the yellow fever vaccine. Not many people had got the Nobel Prize for the vaccination, but some did. So even in 2023, right? But we'll talk about it a little bit later. But the very first was Max Tyler. So he managed to cultivate the yellow fever and attenuate it, similar to Louis Pasteur, so that it got inactivated in the embryos of chicken, as we discussed previously. So the main problem, like the critical medical problem, infectious problem of the beginning as well as the middle of the 20th century was poliomyelitis. And that is something which we are still suffering from, right? We still have the potential to eradicate, but we are very close, so we almost eradicated it. Yeah? But it is known for many centuries and even thousands of years. You can see here, so it was known still from the ancient times, from the Egypt times. So poliomyelitis, that was quite an interesting story that actually it was thought. It was postulated by Simon Flexner, who was at that time the Rockefeller University, he was the president of Rockefeller University, There was the dogma that poliovirus infects nerve cells. Okay, so only neurons, and can grow only in neurons. And that is why, like, in order to make the vaccine, you need to have some model, right? You need to work with something. You need to collect some material. You can multiply, but people are trying. It didn't work. Also, they were struggling for years. Then at some point, there was John Anders, Friedrich Robbins, and Thomas Weller, who were working on a completely different virus. And they were working on the on the epithelium of on the epithelial cells. And then they just used the poliovirus in order to just have a control. Okay, since polio is not growing on the epithelial cells, okay, they use it as control, as a non-growing control, so to say. And they were really surprised to find out that it actually grows really well. Yeah. So, and that is the way how dogmas should be really trusted unless you really prove it. So that is just one of the examples. Yeah. So something which teaches us to be careful and to criticize our own results as well as criticize the results of some really big names, whatever. So you need to see the data. You need to see like what is really going on and in which context. Okay, because they didn't get the Nobel Prize for development of the vaccine, but they got the Nobel Prize for the discovery of the way to cultivate polio, because that was not the key. Then it was just a matter of some short time, and then the polio vaccine was developed. Okay, so then let's go further and talk about the okay. So next one was Joshua Lederberg. So he was very young. He was 27 when he got his Nobel Prize for transduction of genes by phages, so he found that there is the genes can be transduced by phages, and then Macfarlane Bonnet, he was truly amazing. He's from Australia, and the Nobel Committee it was clear that he should have gotten his Nobel Prize, but they didn't know a lot because he was so, his interest was so broad. So he was looking at several different viruses. He got several different discoveries. He made several discoveries in several different viruses about immunology. For example, he was the one who was related about the clonal theory of the B cells. That there is those B cells which encounter the antigen, they got multiplied, and then they elicit this immune response. He was also the one to actually postulate the principle of lysogeny. We'll talk about the Jackson, but he didn't get Nobel Prize for that. So he was not recognized for that, but he was truly a visionary. So actually, Lvov, who we were talking about Lvov before, he coined several terms like lysogeny or capsid or capsular. He was also talking about this DNA or RNA, which should contain within the virus. So Lvov got his Nobel Prize for the discovery of the lysogeny. And that's André Lvov from France. So what is lysogeny? Let's talk about that. So basically, not for all viruses, is characteristic not for all viruses, but for some.
right? So sometimes the viruses go through this living cycle. So what is the living cycle? Means like, okay, so they go through lysis, so they infect the cell, they multiply, and then they just switch all of the machinery of the cell to produce their own protein, their own nucleic acids, and then they kill this. So they lyse the cell. That's why it's called living. Okay. It's in some cases, it's quite often, this strategy is not very productive, because if you kill all of the hosts at some point, you don't have anywhere to notify, but they are obligated parasites. So you need to leave some of the hosts in order to replicate further. So that's why at some point they can switch to the lysogenic cycle. So lysogenic cycle, you see on the bottom, right? So where the virus or the phage remains silent. So it does not replicate, or it replicates only like just as often as the cell replicates. Okay, so it can insert itself to the genome, or it can remain as an edisome. Edisome is like plasmid, and it can stay there in a dormant state. Okay, so this kind of virus, it is within it could be called pro-virus, or it could be called prophage. And at that point, it is dormant until there are conditions that it can switch back to the release. Okay? And that is a strategy to let the host survive, to let enough host in order to have enough generations later, so to not exhaust this niche from the host, because it's obligatory to have some cells to replicate them. Right, and how can this switch be occurred? Okay, so they could be voting. Okay, so they are viruses are voting. Basically, there are several viruses which are infecting the cell, and they are kind of having the mechanism to vote. Okay, so I vote for lysogeny, I vote for lysogeny. Okay, the virus stays alive. And then one is voting, and I'm voting for lysis. Okay, and then the whole process starts, and then the virus goes into the living cycle and kills itself. Or there could be some peptide. There could be some peptide which is important for glycogen. In a small concentration, so the peptide is inactive, and that's why the virus infects the cell, and it gets always goes always to the peptide all the time. But if there are too many viruses which are infecting the cell, then there will be much of this peptide, so it will accumulate, and this peptide is critical for switching to the lysogeny. And then, if there are too many viruses, this peptide will dictate that this virus will go to the lysogeny, and it will not kill the cell. So it will preserve the cell, because there are already too many viruses in the environment, so it's not needed to produce more. And so that is one of the ways. We will talk later about the different other ways how to about switching those. Okay, so let's go further and talk about finally, Peyton Rouse had discovered the Rouse sarcoma, so the first solid tumor which was discovered to be induced by a virus, which was discovered in 1911. And he got his Nobel Prize in 1966. And this virus had got Nobel Prize 2 times more. Later, we will see that. Firstly, it was David Baltimore and Howard Terman who discovered in the same virus that
there is a reverse reverse polymerase. Okay, so that was discovered in this Rouse sarcoma virus. Okay, that was the second time this virus got awarded the Nobel Prize. So Renato Dudek also got the Nobel Prize for discovery of the mechanism of replication of other viruses. The third time, this virus had got been awarded the Nobel Prize when the oncogenes were discovered. Because it was thought that, okay, so those oncogenic viruses, they are oncogenic per se, and so they are just from their properties, that's what makes them oncogenic. It turns out it's not true. It turns out that there are specific genes, specific proteins which are critical for these oncogenic properties, and this was discovered by Michael Bishop and Harold Varmus. They were awarded in 1989. By the way, David Baltimore was also very, he was around 30 years old, and he also developed the nomenclature of viruses, which we will actually follow. So we will talk about DNA viruses, RNA viruses, and he suggested to name them based on the strand. So if we have the DNA, so here is a 3' 5', okay, and here is RNA. So that if these are the RNA will be called plus RNA. Okay, so the template DNA from which this RNA and this size will be minus RNA, and the opposite strand will be called plus. So by that, it is really helpful to make a classification of viruses, because some of them they tend to have as their genome the minus strand, the others plus strand, and then that really is something, as well as same is true of for RNA viruses. So we will talk about that later. Okay, so in 1969, Max Dalwood, Hershey, and Luria, so they discovered recombination that was also done on viruses. And it's important that Hershey, together with Marco Chase, in 1952, proved that during the infection of bacteriophage T4, it was DNA and not some other components like proteins that were really causing the infection. So they have proven before Watson and Crick that DNA was the key for the transfer of the genetic material. Actually, there were, even before that, there were several other observations. So before Watson and Crick postulated the dogma of molecular biology, it was Chaga who discovered that there is an AT and GC pairing. So it was not Watson and Crick who postulated that. And also in 1948, it was also discovered that actually the germ cells have half a genetic material than the somatic cells. So that should also have ring the bell, right? So that it is probably that should be the genetic material. So they should have guessed, but they did not. So in the end, in 1953, it was Watson and Crick, who managed to discover the DNA, the structure of the DNA, and they got their Nobel Prize later. In Baruch Blumberg from Australia. So he was an immunologist, and he managed to discover the antigen of the hepatitis B, and that was the discovery called hepatitis B. And the Gajdusek, he lived in Africa, and he managed to prove that the Kuru disease, which is caused by rhinos, was actually transferred by monkeys and within the brain, and those people who were eating the brains of those monkeys, they got the disease, but he didn't know the structure. So that later, much later, the structure of the prions was discovered. And here we will talk about the Stanley Personnel. So he got this Nobel Prize for the prions again, right? After the Gajdusek. So at that time, he proved that actually prions are those protein agents which don't have any capsid or envelope, they don't have any nucleic acid, but they can infect by just this not regular conformation, like this weird conformation of those proteins per se can transfer to other proteins, and this kind of infection of this by this conformation, this wrong conformation, is per se an infection. Okay, so that is the prion disease, and the field is actually really growing, and there is now a term of prion-like disease. So it's believed that Alzheimer's disease with the tau protein as well as the beta amyloid as well as Parkinson's disease with the alpha-synuclein, those proteins, they have this kind of prion-like structure. So this wrong conformation which can infect further cells and just spread, and it is believed there is this rough theory of Parkinson's disease initiation that it actually comes from some infection agent within the gut. So within our intestine, those wrong conformation being it starts to spread through the nervous bundles towards the brain, towards the midbrain, and then infect the substantia nigra, pars compacta, where those cells, the developmental cells, start to die because they accumulate those alpha-synuclein, which is having this kind of wrong conformation. And there are many examples and many data which confirm this. And just several weeks ago, there was a paper, I think it was Nature, that it was in New England Journal of Medicine. They found that when there was no recombination technology yet, people had injected the somatotropic hormone to people who were very small people who had a lack of this hormone. Like Lionel Messi, he was also not having enough of this hormone, so he was talking to that for in order to make sure that he got his therapy. So that little bit high, a little bit power. And right, so what people were doing, they were actually taking the sample from the pituitary of dead people in order to have this kind of somatotropic hormone and the growth hormone, basically, and inject it to those diseased people. And they found that it turns out that at that point, some of those people from little dead people, they had Alzheimer's, and it turns out that this was kind of an infection. So they then transferred Alzheimer's by those injections. So there was very convincing data. Also, telling in favor of this kind of prion-like diseases and this theory of spreading of those prion-like phenotypes just based on the conformation. Yeah. not really virus, yeah, but still we will be happy to discuss that because it is also, I think, also very interesting. Okay, there was a second Nobel Prize for the prime disease, and the third one was actually given in 2002 to Kurt Wittrich, who managed to use genetic resonance of those to determine the structure of the prions, like how really do they form those kind of weird infectious structures. Okay. Paul Berg, of course, like this, it's very important, the viral vectors. So all of these T promoters, T7 promoters, M13, and many kind of origins of replication, we don't even think about that. Like everything what we use, all those different ways of recombination or the fields and on, we don't need to think about that. But those, most of them, they would derive from viruses. Okay, so the capsid structure principles, which was developed by Aaron Klug, and he got his Nobel Prize in 1982 for crystallization of the viruses and these icosahedral capsids. So that was really critical to determine the structure of those, and it was important that unfortunately, so the collaborator of Watson and Crick, who got the Nobel Prize for this dogma of molecular biology, she was actually Aaron Klug's supervisor, and she was actually interested in those capsids in those viruses. So she should have got this Nobel Prize twice, but she didn't do that because she actually died from cancer. Right. So let's go further and just talk about the Gertrude Allen, who was working in George Hitchings' laboratory, and she was interested in developing the antiviral agents, and she was trying to find something which was like not so similar to the host, and in terms of the herpes viruses, she found that the thymidine kinase in herpes viruses was a little bit different structurally, as well as on the molecular level. It also had different substrate preference. So she developed a cyclovir. So there was the first antiviral drug, and that basically could convert this cyclovir into this toxic compound that could kill the cell. And it was converted only by this thymidine kinase of the virus, but not by our own thymidine kinase, because actually even thymidine in the herpes virus is different, so it is modified. Some viruses do this in order to evade all these kind of antiviral defense mechanisms. For example, some of the bacteria, they use restriction enzymes. That's why some of the phages they modify the phases in order to hide from those kind of diseases. So it's kind of its arm race. Yeah. So you have immunity, you have anti-immunity, and like that's how we hosts as well as viruses we co-evolve together. We live together. Okay. Richard Roberts and Philip Sharp, so they developed splicing. So they discovered splicing it was done in adenoviruses. Then Doherty and Zinkernagel, they discovered T-cell immunity against lymphocytic chronic lymphitis virus. And then HIV, that was a very important discovery. So Baron and Sinussi as well as Montagnier. And Harald Stockhausen also discovered that the different types of papillomaviruses, 12, 18, 31, were involved in the genesis of the cervical cancer, and